Hello everyone, my name is Visalhounuo. In this video, I'll be explaining on triple sugar ion agar test. Triple sugar ion agar test is used to determine whether gram-negative bacilli utilizes glucose and lactose or sucrose fermentatively and produce hydrogen sulfide. It contains 10 parts of lactose, 10 parts of sucrose, 1 part of glucose, and peptone. Phenol red and ferrosulfate serve as an indicator for acidification of medium and H2S production respectively. Glucose is utilized first by a fermentative organism and the entire medium becomes acidic, yellow in color, in 8 to 12 hours, but remain acidic even after 18 to 24 hours incubation period because of the presence of organic acid resulting from the fermentation of glucose under anaerobic condition in the bud of the tube. The slime reverts to alkaline state that is indicated by red color as the fermentation products gets oxidized to carbon dioxide and water. And peptone in anaerobic condition, the slime undergoes oxidation, releasing alkaline amines. Phenol red in alkaline pH turns red, while in acidic pH turns yellow. If the organism ferments glucose but does not ferment lactose and or sucrose, then the slime becomes red and bud remains yellow within 18 to 24 hours. If the organism is in addition to glucose ferments lactose and or sucrose, the fermentation products forms on the slime will more than neutralize and the alkaline amines rendering the slime acidic yellow in color, provided the reaction is red within 18 to 24 hours. If the organism is non-fermenter, instead of sugars, peptone is utilized as an alternate source of energy under aerobic condition of the slime, which makes it alkaline indicated by the red color, while there is no change in the color of the bud. Reaction in the triple sugar ion should not be read after 24 hours of incubation because aerobic oxidation of fermentation products forms lactose and or sucrose will occur and the slime will eventually revert to alkaline state. The formation of carbon dioxide and hydrogen is indicated by the presence of bubbles or cracks in the medium or by the separation of the agar from sides or bottom of the tube. The production of hydrogen sulfide requires an acidic condition and is indicated by the blackening of the bud, the medium in the tube. Procedure. With a sterilized, straight inoculating needle, touch the top of a well-isolated colony. Inoculate triple sugar ion agar by first stabbing through the center of the medium to the bottom of the tube, then streaking on the surface of the agar slant. Incubate the tube at 35 degrees Celsius for 18 to 24 hours and observe the reaction. Observation. Alkaline slant and no change in the bud, or alkaline slant with alkaline bud is glucose, lactose, and sucrose non-fermenter. That is, they appear red in slant and red in bud. Alkaline slant or an acidic bud, that is glucose fermentation only, appearing red slant and yellow bud. Acidic slant and acidic bud, that is glucose, lactose, and or sucrose fermenter, that is with a yellow slant and yellow bud. A black precipitate in the bud indicates production of hydrogen sulfide. Hydrogen sulfide reacts with ferric salt to produce black precipitate of ferrous sulfide. Bubbles or cracks in the tube indicates the production of carbon dioxide or hydrogen. Drawing of circle around bud indicates that gas is produced by glucose and sucrose or sucrose, lactose and glucose, sucrose and lactose fermenter. Examples. In case of acidic slime and acidic bath with production of gas and no production of H2S. Example is Escherichia, Klebsiella, and Enterobacter. If an alkaline slime with acidic bath with no gas production, no H2S production, then example is Shigella and Serratia. If an alkaline slime with acidic bath with gas production and H2S production, then example is Salmonella and Proteus. If an alkaline slime with alkaline bath with no gas production and no H2S production, example is Pseudomonas.